Good morning, everyone. I'm one of the theme leads for Metabolic Physiology, and it is my very great pleasure to introduce this year's Michael de Burgh Daly Prize Lecturer. Michael de Burgh Daly was Professor of Physiology and Head of the Department of Physiology at St Bart's Medical College from 1958 until his retirement in 1984. His research was primarily concerned with the carotid body chemoreceptors and baroreceptors. Through careful experimentation, he contributed much to our current understanding of the mechanisms underpinning cardiovascular and respiratory reflexes. He was an active member of the Physiological Society throughout his career, and over 2 periods served as co-editor of the Journal of Physiology. Following his death in 2002, the Society endowed the biennial Michael de Burgh Daly Prize Lecture on the topic of autonomic control. The first lecture being delivered in December 2002 by Professor Janice Marshall of the University of Birmingham. This year's lecture will be delivered by Professor Fiona Gribble. Fiona is Professor of Endocrine Physiology and Deputy Director of the Institute of Metabolic Sciences, University of Cambridge, and an Honorary Consultant in Clinical Biochemistry at Addenbrooke's Hospital. Fiona's work concerns the cellular physiology of gastrointestinal endocrine cells and the incretin axis. In particular, her group uses an approach based on electrophysiology and fluorescence imaging to study the events involved in stimulus detection and mechanisms of hormone secretion and action, work which has enormous implications for the identification of novel anti-obesity and anti-diabetic therapies. Fiona has received many honours, including the Lister Prize, the Minkowski Prize, and the R.D. Lawrence Prize Lectureship. And in 2015, she was elected a Fellow of the Academy of Medical Sciences. Fiona, many congratulations on this award. We're delighted to have you here with us today, albeit virtually, and we're very much looking forward to your lecture, titled The Gut Endocrine Axis in the Control of Metabolism. Fiona.
Thanks very much, Andrew. And thanks very much for it's a great honour to be given this award, and I'm delighted to be able to give the lecture. It's very strange to be giving it from my office in my house and not to be able to look any of you in the eye and see if I'm boring you rigid. So hopefully I'll take it on trust that I'm not boring you rigid. So what I'm going to talk to you about, as Andrew said, is the gut as an endocrine organ, and in particular the role that the gut plays in the control of metabolism. So the gut actually produces around 20 different bioactive peptides, which I've put a number on this figure. When I was first introduced to gut endocrine physiology as an undergraduate, we learned about what these hormones do in terms of controlling gallbladder contractility, exocrine pancreatic secretions, and gut motility. And the focus was all around how the gut actually controls nutrient digestion. What's become much more apparent and made it a much more interesting topic is that not only do these hormones control how the gut digests and absorbs food, but they also control how the body deals with the nutrients once they've been assimilated into the body. So it controls how the rates of insulin secretion, for example, and as we heard in the symposium on GLP-1 yesterday, how the body detects food intake to control appetite. So it's not just the food absorption, it's also what is done with the food and how the body knows that we've been eating. And the hormone that we've been working mostly on, although we've diversified quite a lot, is this hormone called glucagon-like peptide 1. It is important for controlling gut motility and therefore rates of nutrient digestion and absorption, but it has these important effects in controlling insulin release and appetite. And for those of you who don't know GLP-1, it comes from this pro-glucagon peptide, which is a fairly long propeptide that contains the glucagon sequence that we all know that's produced in pancreatic alpha cells. And pancreatic alpha cells have prohormone convertase 2, so they cleave this pro-glucagon sequence to release glucagon, but they don't release active GLP-1, GLP-2. They leave those in a longer C-terminal sequence. However, these endocrine cells in the gut called L cells, they have prohormone convertase 1,3, and that cleaves out GLP-1 and GLP-2 from this long pro-glucagon sequence. And it actually leaves glucagon in a longer sequence called oxyntomodulin. And oxyntomodulin has some but reduced bioactivity on the glucagon receptor and some bioactivity on the GLP-1 receptor, but the important peptides we think that come out of these the gut endocrine cells are these GLP-1 and GLP-2 peptides. And one of the major actions that they have is in controlling insulin secretion. So even before GLP-1 had been discovered, it was known that the gut plays an important role in regulating insulin secretion by this effect known as the incretin effect. And this is a particularly good demonstration of the incretin effect, an experiment done by Michael Nauk. And what he did was to take a group of healthy human volunteers, and he brought them in on one day and gave them an oral glucose tolerance test. And he measured the plasma glucose over time, and he measured the plasma insulin. Then the clever thing was that he brought them back on a second day, and he gave them what's known as an isoglycemic intravenous glucose tolerance test. So the isoglycemic means that he mimicked the plasma glucose profile to that person's profile they had on the day when they had the oral glucose. So he had matched plasma glucose profiles. But when the glucose was given intravenously, the people only produced about half the amount of insulin. So the pancreas should have seen the same plasma glucose concentration on both days, but it was responding differently by producing more insulin on the day when the glucose was taken orally. And this difference in insulin release between the oral and the intravenous day is known as the incretin effect. And something that Michael Nauk also showed in this same study was that people with type 2 diabetes have a bit of a reduced incretin effect. I won't come back to this. We don't think that's the cause of diabetes, but probably partially a consequence of diabetes. And the physiology of this is that if you give the glucose intravenously, glucose acts on pancreatic beta cells directly. It closes KTP channels, which causes the cells to depolarize, and you get voltage-gated calcium entry that triggers insulin secretion. But if the glucose is taken by mouth, then as the glucose is absorbed through the gastrointestinal epithelium, these hormones, GLP-1, together with the partner hormone called GIP, are released at the same time. And these, when they target the pancreatic beta cells, act on G-protein coupled receptors, which elevate cyclic AMP. And the consequence is that cyclic AMP synergizes with the signal coming from the glucose and causes this dramatic enhancement of insulin release. And then something else that really made this enter the field of interest of academia and industry was some more experiments done back in the 1990s after GLP-1 had been discovered. And this is an experiment again done by Michael Nauk, where he infused GLP-1 into people who had type 2 diabetes. He gave them a 4-hour infusion of GLP-1, and during that time, the plasma glucose dropped from the diabetic range, where it started about 10 millimoles per litre, and it dropped down into the normal range, which is more around the sort of 4 to 5 millimoles per litre. And it did so together with stimulating insulin secretion. So likely it's this insulin secretion that is driving this lower blood glucose. So actually my first foray into research was, I was a young junior doctor at the time, and I joined the group of Robert Turner to do some experimental medicine in humans. And we had, following on from what Michael Nauk had done, said, what would happen if you gave an overnight infusion of GLP-1 to people with type 2 diabetes? So this is now an overnight GLP-1 infusion. And the same patients came back on 2 other days where they either had a saline infusion overnight or an insulin infusion overnight, which we graded to give the same sort of plasma glucose drop as the GLP-1 had. And then the following morning, we gave them a hyperglycemic clamp, and we measured their insulin response. These 2 lines at the bottom show the saline and the insulin control arms in the diabetic group, and you can see they had not much response to the hyperglycemic clamp. The dashed line here shows a control group who we just gave a clamp to. So you can see that they have much more. It's the first phase response of insulin secretion, and the second phase, much more than the diabetic group. But when we gave the diabetic group the GLP-1, they effectively produced a normal insulin secretory response to this hyperglycemic clamp, And all measures of beta cell function that we could measure were actually restored by the GLP-1. So this was actually part of a growing body of evidence back in the 1990s that maybe you could use and target GLP-1 to treat type 2 diabetes. It's not quite as simple as just giving GLP-1, however. GLP-1, if you look at plasma levels after a meal, they rise quickly and then they fall quickly. And part of the fall is because the GLP-1 that's released from these enteroendocrine cells is very rapidly degraded by dipeptidyl peptidase 4. Actually, circulating GLP-1 has a half-life of just a few minutes before it's inactivated. So giving GLP-1 itself is not a useful therapy. And the therapies that actually were developed were inhibitors of DPP-4. So we now have these on the market. It's an oral treatment, and they block this degradation of the active GLP-1 to the inactive GLP-1, or modifications of GLP-1 that make it more stable in the circulation. So there's a natural peptide called exendin-4 that comes from the Gila monster that is stabilized against DPP-4. And pharmaceutical companies started changing around the GLP-1 sequence. So they modified it so it could be DPP-4 resistant, and so that it's stabilized in the circulation, and even so that it can be administered orally rather than by injectables. And probably the best in class one is this one from Novo Nordisk called semaglutide, which has modifications to make it DPP-4 resistant, and this fatty acid side chain that helps to stabilize it by binding to albumin. And semaglutide, along with some others, you can now give actually as a once weekly injection for people with diabetes. And the effects are very good. So this is looking at, it's a semaglutide trial over 2 years. So it's once weekly injections of semaglutide. And if you look at the HbA1c, for those of you who don't know the HbA1c range well, an HbA1c of sort of 8.5 to 9 is a quite uncontrolled diabetes. And less than 7 is where we're aiming for diabetes control with people with type 2 diabetes to be. So what you can see is that semaglutide brought the HbA1c down to pretty much towards a range where you would want the diabetic control to be. Although ideally you might have it further down. Actually, most drugs we have for diabetes can bring the HbA1c down by about a 1 percentage point. So semaglutide is probably a bit better than many, but it's not bringing it completely down into normal range, we should be clear. The other thing that became evident when people started being treated with GLP-1 agonists is that it also affects body weight. And we did again hear more about this in the symposium yesterday as to how this might work. But this is a 1 year trial of a higher dose of semaglutide in people who didn't have diabetes, they just had obesity. And you can see a 15% drop in body weight in the group treated with semaglutide. So we really do now have GLP-1 based treatments that we can use both for obesity and for diabetes. But what I want to talk to you more about is the basic physiology of the gut. And I've shown you this figure already of the incretin axis. I actually, after I did my clinical research on GLP-1, I moved and did a PhD with Fran Ashcroft in the University of Oxford on KATP channels in pancreatic beta cells at the time when these channel subunits had just been cloned. And when I set up my own research group in Cambridge, which I actually did with my husband, Frank Ryman, and we've been running it together ever since, we decided to look at these cells that make GLP-1. So they're potentially analogous to the pancreatic beta cells, but they sit in the epithelial lining. And very little had been done with studying how they work back in the around 20 years ago, because they sit embedded in the epithelium and they make up about 1% of the epithelial layer. And if you just take the epithelium off the gut, you don't know which is a cell that produces GLP-1 and which is an enterocyte. But they do respond to ingested glucose. So we thought, perhaps they're similar to pancreatic beta cells in the way they work, but there are differences. So Whereas plasma glucose is a strong stimulus of insulin release, it doesn't stimulate GLP-1 release. So this is plasma GLP-1 in response either to oral glucose or IV glucose. And you can see that actually the intravenous glucose doesn't work. It's just when you give it to this side. So it's a slightly curious cell type. And these cells are derived from crypt stem cells, which divide continuously and they differentiate. Most of them form enterocytes, but some of them form these endocrine cells. Some of them also form the goblet cells and Paneth cells. And they're produced in the colonic crypts, so these cells turn over regularly. They move up out of the crypts into the villi. And in the small intestine, they have a lifespan of about 3 to 5 days before they're extruded from the villus tips and lost into the gut lumen. And they sit resident in the epithelial layer. So this is my depiction of an enteroendocrine cells. And I used to say that they had this apical surface facing into the gut lumen, and that isn't this ideally placed to sense what's going past in the lumen and provide a signal to the body that says what's in the gut lumen. As I'll say sort of as I go along, I don't think this is actually quite the right model anymore. But how do you even study it? So we started off in the field studying a cell line called glutag. It's an excellent cell line, but we were criticized right from the beginning of, well, why do you study a cell line? How do you know that a cell line is actually a good model for native L cells? So then we moved into transgenic mouse models. And we've made a number of mouse models where we've labeled enteroendocrine cells. So this one, I'll show the example of, we use the pro-glucagon promoter to drive expression of a fluorescent YFP derivative in a transgenic, a back transgenic mouse model. And then we develop protocols to take the gut epithelium out of a live adult mouse and to culture it. And here you can see the white cell is a fluorescent cell, so that's a cell that's making pro-glucagon. The rest of the cell are other epithelial cells that we can't identify. But this allowed us for the first time to purify the cells, to put patch clamp electrodes on, to monitor intracellular calcium, and to work out how these cells are functioning. The field has moved on even more since then. So a group in Utrecht, led by Hans Clevers, worked out how to keep these primary cultures alive in living culture, in a three-dimensional culture by providing stem cell niche. So you can now make organoids out of these mouse models. So this is one actually where the fluorescent sensor here is a cyclic AMP sensor just expressed in the L cells. And the most recent iteration of this model, now we've taken human ileal organoids, so they're from human biopsies, and then we use CRISPR-Cas9 to insert, again, a yellow fluorescent protein into the native proglucagon locus. So all these cells that appear as green are human L cells being generated in this self-regenerating human organoids in culture. So then you can take these human L cells and do lots of analysis on them. You can digest this up to single cells, purify the cells out by their fluorescence to purify the fluorescent L cells away from the non-fluorescent cells. You can do peptide mass spec, for example, to see which peptides are being produced in the cells. You can run RNA sequencing to see which transporters and ion channels and receptors are being expressed in the cells. Down the bottom here is just our validation to show that the venous, this yellow fluorescent protein, is expressed in the cell that makes the proglucagon. And on the right are some examples of sort of recordings that we make from these cells. So if you put patch clamp electrodes onto the cells, you can see that they fire action potentials. These are overshooting action potentials. I haven't put a scale, but they overshoot to around plus 40 millivolts. We found these also in mouse primary cells, and we found them in glutag cells. And in general, there are some stimuli that will enhance electrical activity, like glucose here. This is a recording, just for example, to show this. You can load the cells with Fura2 and do calcium imaging. We've also made some of these where we express a GCAMP directly into the cells of interest. But you can apply your test agents and see what calcium responses you get. Or you can culture these organoids and look at which stimuli trigger GLP-1 secretion. And so you can build up a picture of how the cells work. And the particular questions we've been asking over the years is what and how do enteroendocrine cells sense? So which receptors are expressed by the cells, which pathways trigger secretion, and where are the receptors localized on the cells? So if we start with the simple case of glucose detection, L cells do actually have GLUT2 and KHP channels, similar to what we find in a pancreatic beta cell, but KHP channel inhibitors, whereas they stimulate insulin secretion, they don't increase GLP-1 in humans. And if you block GLUT-mediated glucose uptake in the cell line, you can block glucose uptake and measure that, but that doesn't actually block GLP-1 secretion. And interestingly, people who've done perfused intestinal preparations using an intact piece of gut have found that non-metabolizable substrates of this sodium glucose cotransporter will trigger GLP-1 secretion. And actually, we've done a lot of work over the years, and we've knocked out SGLT1 and inhibited SGLT1. And we think that SGLT1 is actually acting as a glucose sensor. So what this protein is a glucose transporter that has an obligate coupling of 2 sodium ions to every glucose molecule that enters the cell. And you can get a magnitude of a few picoamps of current generated by this transporter at high glucose concentrations. And we've shown that that is enough to drive a small membrane depolarization, which can pick up and trigger action potentials and then trigger voltage-gated calcium entry. And that then leads to GLP-1 secretion. And I think this is quite clever by the L cell, because what it's actually doing is linking the GLP-1 signal that is generated actually not just the amount of glucose that's floating around in the gut lumen, but the amount of glucose that's actually being absorbed into the bloodstream at the time. Because what the pancreatic beta cell wants to respond to is the amount of glucose that's entering the bloodstream, and not just what is still sitting in the gut waiting to be absorbed. So that's glucose, but actually lots of different components of a meal will actually trigger GLP-1 secretion. So dietary components, so products of protein digestion, products of fat digestion, bile acids, microbial fermentation products like short-chain fatty acids and lipopolysaccharides. There's also some link with the neurohormonal system. So arginine, vasopressin, and angiotensin will trigger enteroendocrine secretion. We think probably this is not necessarily GLP-1 that's important, but maybe PYY, because PYY will inhibit fluid secretion from the gut. So maybe this is part of a volume regulation role of the gut endocrine system. And many of these receptors, the ones that I've put circles around in particular, are actually being actively looked at by industry as a potential targets for targeting drugs to the enteroendocrine system. This is so we did a lot of characterization in mouse over the years. The human L cells look fairly similar in terms of their receptor expression. So this is from the RNA sequencing data, the receptors, key protein coupled receptors that are highly expressed in L cells compared with non-L cells from humans. And it's the same body of receptors that are coming again up in the human L cells. So there's receptors for long-chain fatty acids and monoacyl glycerols, for bile acids and amino acids and short-chain fatty acids. And there's quite a number of others hidden in the gaps there that I haven't highlighted, but many of these are actually involved in nutrient detection. And wouldn't it be nice if these receptors were sitting on the apical membrane and actually detecting the nutrients as they come past, potentially allowing you to develop a non-absorbable drug that would target these receptors, have no side effects because the drug isn't being absorbed. You'd have a nice clean way to trigger GLP-1 secretion. That's what we'd hoped. In practice, that's not what we and others have found. So actually what we're all finding is that the receptors are localized on the basolateral side of the cells. So bile acid sensing, for example, if you block the apical sodium coupled bile acid transporter, so the bile acids can't get across the epithelium, they won't trigger secretion, and the receptor seems to be only accessible from the basolateral side. That is G protein coupled bile acid receptor, also known as TGR5. Similarly, long chain free fatty acids, we think maybe this transport it's using is chylomicron formation with release of long chain fatty acids again on the basolateral side, and targeting these free fatty acids, again, localized and accessible largely from the circulation side. And similarly, also now we're discovering for amino acids, likely being detected by the calcium sensing receptor. So the sort of experiments people are doing to identify this, we've used Ussing chambers to measure it. But if you use a piece of perfused intestine, you can perfuse agonists of these receptors either from the luminal side or from the vascular side. And the results of these are that only from the vascular direction will agonists of these receptors actually trigger secretion. So it means that we can't target drugs to the apical side. They need to be absorbable. But I think it's also quite important, again, for physiology, that what the body is doing is responding to what is being absorbed and not just what is present in the lumen. So I think the physiology is clever, but it makes it harder to target with therapeutics. So which of these stimuli trigger GLP-1 secretion? The answer is some of them more than others. So this is secretion from these human ileal organoids. And this is agonists against this bile acid receptor are strong stimuli of secretion. Here you can see arginine vasopressin, GPR119 agonists. And some of these receptors are GS-coupled. GS-coupled receptors seem to be particularly good stimuli or good receptors to target to trigger GLP-1 secretion. Free fatty acid receptors and angiotensin receptors are GQ coupled. And this is actually a superagonist we've used of the free fatty acid receptors. In general, we find in vitro that GQ coupling is not quite as strong as a stimulus. Once you get to these coupling mechanisms downstream of the receptors, they're probably pretty similar in enteroendocrine cells to other stimulatable cells. So general coupling to IP3 mediated calcium release, although there is some evidence that free fatty acids might start opening TRP channels. Exactly which channel it is seems to be a bit species dependent and cell line dependent. So we're not quite certain about the universality of this TRP channel involvement. And the GS-coupled receptors are targeting adenylate cyclase, elevating cyclic AMP, part of which is enhancing the secretory response, but part of it is also increasing cellular excitability and making the cells more responsive to some of these other depolarizing stimuli. So the question we're facing is, could you actually target L cells to treat diabetes and obesity? And the reason that we're hanging in and thinking that maybe you could do this is because of the effects of bariatric surgery. So bariatric surgery are operations that change usually the rerouting of nutrients in the gut and is used to treat obesity originally, but increasingly more diabetes together with obesity. And this is the effect of different bariatric procedures on weight change from the Swedish obesity study. And this is over 15 years. You can see it's effective and maintains effectiveness. And these are data, again, from the Swedish obesity study showing the incidence of developing new type 2 diabetes. And you can see it's lower in the bariatric procedures than in the control group. And even mortality, over the first 2 years after you've had bariatric surgery, mortality is higher in the surgical patients, and this is the post-operative and perioperative mortality. But after 2 years, the surgical patients do much better in mortality. And the most common operation or the most effective operation is a Roux-en-Y gastric bypass surgery. So here the jejunum is transected. The bottom part is pulled up to join up to a small stomach pouch, and the biliary and pancreatic secretions are brought down a duodenal loop and joined a bit further down. An operation that's increasingly being done now is a sleeve gastrectomy, where effectively the stomach as a sac is being turned into a stomach as a tube by taking off the majority of the stomach. And data from obese people who've had a Roux-en-Y gastric bypass show that even after a week after surgery, plasma glucose levels are lowered. And that whereas these patients who had been type 2 diabetic before the operation have looking close to a non-diabetic type phenotype. And something that is dramatic and repeatable is that the GLP-1 levels before surgery, they're relatively low when you look on this scale, but after surgery, they're going up to very high levels. And it happens soon after surgery. The model that we've been using to study this is a group of lean patients who've had a gastrectomy surgery because of an inherited form of gastric cancer. And they have an operation that's very similar to this Roux-en-Y gastric bypass, except that the stomach is taken out completely. But then the rerouting is similar to bariatric surgery. And like the obese bariatric patients, these gastrectomy patients have very high postprandial GLP-1 and PYY levels. And we've used these human models to ask a few questions, one of which is, are the gut hormones, these changes, these very high levels of GLP-1 and PYY, are they responsible for any of the metabolic benefits of surgery? And what is the mechanism for this enhanced release of GLP-1 and PYY? So if I take that first question first, what we hypothesized was that so this is I haven't showed you, this is the plasma glucose and insulin levels in the lean gastrectomy group. You can see they have very high plasma insulin levels, and actually many of these patients actually complain that if they have a sugary meal, they actually have to lie down afterwards because they start to feel a bit lightheaded. And we found a number of them who were dropping into the hypoglycemic range after a 50-gram oral glucose tolerance test. So we made the hypothesis that maybe the driver of this high insulin was this combined glucose signal arriving at the same time as a very high GLP-1 signaling and targeting the pancreatic beta cells with these synergistic pathways. And we used exendin-9, which is a peptide antagonist of the GLP-1 receptor. And we brought a group of these patients in on 2 occasions. And on one day, we gave them a 50-gram GTT together with a control infusion. And on the other day, they had an exendin-9 infusion. And you can see that when they had the exendin-9, the plasma insulins had fallen back down effectively to the normal range, and they no longer had the hypoglycemic episodes. So we conclude from this that these high levels of GLP-1 that you get after bariatric surgery are high enough actually to target the pancreatic beta cells, and provided the pancreatic beta cells are themselves have enough function to remain, then that will trigger more insulin secretion. There is a problem with people who've had type 2 diabetes for many years, which is that they just don't have enough beta cells to be able to respond to this GLP-1 signal, which is why I highlighted that you might need enough functional beta cell mass or functional beta cells to be able to respond to the GLP-1 signals for this to happen. There is much less evidence, however, that the GLP-1 is responsible for the appetite suppression that you get after bariatric surgery. So we don't think it is just GLP-1. We think that that's driving the bariatric surgery improvements. We think it's other peptides as well. This is a mass spec analysis we've done of the mouse GI tract, and we've done it also in humans. So we've taken the mouse GI tract, split it into 5 centimeter intervals, and done a peptide mass spec analysis. And you can see all the different hormones that are produced in the gut. You can also find, when we do the same from these purified L cells, from these organoids, even though we purified the cells on the basis that they were producing pro-glucagon and therefore making this fluorescent protein, they also contained other peptides. So peptide YY and neurotensin came up in the same population. So this actually builds on the picture we've been developing over years, which is that individual enteroendocrine cells can produce one or several different hormones, and it's not as specified as the classic literature would say. It's not one hormone, one cell, and one cell type. There's a lot of overlap. But in particular, these hormones that we find that go up after bariatric surgery, GLP-1 and PYY, they're produced fairly low down the GI tract. And it begged the question as to whether or not these elevations of GLP-1 and PYY that you get after bariatric surgery, is this because the altered nutrient exposure that the gut gets after surgery is changing how the enteroendocrine cells are developing? So you're getting, for example, more enteroendocrine cells in the upper gut that are producing GLP-1 and PYY. Or is it because you're delivering stimuli to a different part of the gut and the cells are remaining the same? So to address this, we've actually looked at the enteroendocrine cells before and after surgery to see both at the level of their transcriptome using RNA sequencing and their peptidome by mass spec. So this is an example. This is enteroendocrine cells purified from mice where we've done a bariatric procedure known as a vertical sleeve gastrectomy, and we purified the enteroendocrine cells out by fax from the upper gut, the lower small intestine, and the colon and rectum. And we had 3 mouse groups. We had a sleeve gastrectomy group, a sham group, and a sham group that had been weight matched to the sleeve gastrectomy group. And we did RNA sequencing on these, and what you can see is the cells, they know where they've come out from which part of the gut. So the small intestinal ones separate from the colonic and rectal ones, but they don't separate by whether or not the mice have been operated on or not. So the operation is not actually changing the transcriptome of the cells. And this is an example of one we did in humans. So we took biopsies of these lean gastrectomy patients at the time they were having their jejunum transected anyway for the surgical procedure. So we purified. Actually, for this particular procedure, we just took biopsies at this point, and then we went back by endoscopy to people who'd had the operation. And took biopsies from the same matched region after surgery. And then we did mass spec. We did actually also do some RNA sequencing on the purified cells, but these are the mass spec data. And you can see there's absolutely no hint that cells from the same matched part of the gut had changed their profile of hormones they produced after surgery. So actually, we don't think that the hormonal profile is changing at all. The stem cells know where they came from, and the stem cells still produce the same hormones even after surgery. But what's happening is that the nutrients are being delivered to a different region. So in normal postprandial physiology, nutrient absorption and digestion happens high up in the gut, and you get high stimulation of GIP and some stimulation of GLP-1. But after surgery, the nutrient digestion mostly happens after the pancreatic enzymes and biliary secretions have joined the main tract. So digestion and absorption happen lower down. And then you stimulate many more of these cells that are producing GLP-1 and PYY. What you might notice is that actually there's a big population of cells, even in the colon and rectum, that produce GLP-1 and PYY. And these don't normally see ingested nutrients at all. Even after bariatric surgery, there's probably not that much entry of nutrients into the colon and rectum. And most people think that probably these cells in the colon rectum are normally responding to microbial fermentation products and modified bile acids. But this is a population of cells that potentially could be targeted to treat diabetes and obesity if they would be effective. So we did an experiment where we wanted to look at the effectiveness, the metabolic effect of just targeting enteroendocrine cells in the distal colon and rectum. And we used the fact that this peptide insulin-like 5 is only actually produced in the distal half of the colon and rectum. And we made a mouse model using an insulin-like 5 RTTA that Frank made, that we crossed with a DQ dread reporter, so that if you stimulate this mouse with CNO, you only stimulate the enteroendocrine cells in the distal colon and rectum. And you can show, we showed that elevates plasma levels of GLP-1 and PYY. But what we could also show, so this is mouse, you had to induce the mice, and we fasted them overnight, and then we gave them vehicle or CNO together with an IP glucose tolerance test. And after 2 hours, we refed them. And you can see the CNO treated group had improved glucose tolerance, and they had reduced food intake. We used an antagonistic GLP-1 receptor antibody and showed that blocking the GLP-1 receptor axis abolished this improvement in the glucose tolerance, but it didn't affect the food intake. But administration of a Y2 receptor antagonist that blocks PYY receptors, that abolished the food intake phenotype and didn't block the glucose tolerance effect. So our overall conclusions from this is that even though normal postprandial physiology is involved likely mostly with the upper GI tract, the whole of the GI tract produces these different hormones, and even if you target the hormones in the distal gut, you can have better beneficial metabolic effects. And the final thought really is that could one therefore mimic gastric bypass surgery without doing the surgery? And that is the ultimate challenge for the field. The industrial route, we've already heard about GLP-1 receptor agonists, but also that GLP-1 receptor agonists are not on their own as effective as bariatric surgery. So there's something that you need to do in addition to GLP-1 to get a full effect. So pharmaceutical companies and biotech companies are testing different dual and triple receptor agonists, where they're building peptides that have GLP-1 receptor agonism activity, but they also have dialed in agonism to other receptors, such as the GIP receptors and glucagon receptors. So these drugs are in clinical trials at the moment, and some of them looking quite effective. But also maybe GIP and glucagon and GLP-1 together are not enough. So do you also need to dial in some PYY and some other peptides? Or could you use diet and drugs to target the L cells? And this is still the area that we would like to hope will end up having some therapeutic success, because I've already told you there's an overlap between the different enteroendocrine cell types. So if you could target these enteroendocrine cells, you could potentially then mimic bariatric surgery by directly targeting the cells, by targeting these receptors, and develop a mixed gut hormone response that might be more providing a better mimicry of the post-bariatric type physiology. So I'd finally like to thank obviously all our funders and our research group who have done all the work. Particularly thanks to my husband, Frank, who's been co-running the group with me for the last 20 years. This is our current group I've shown here. I haven't got space to add all our many former LAMB members, many of whom did the experiments I've shown you. We actually, in our group, we have a part of the group is focused on the targets of gut hormones in the central nervous system. We have a branch that is working on developing mass spec methods to measure gut peptides, and these are our group at the moment too, measuring the gut and how it responds to stimuli. So thank you all for listening. I hope I didn't bore you, and if any of you are still out there, it's been a pleasure talking to you this morning. Thank you very much.
Well, Fiona, what a tour de force. Thank you for taking us through so many intricate and informative studies, and for the exciting preview of where this work is heading. I'm sure that our audience, wherever they are, is showing their appreciation right now, and there's lots of very positive comments I can see in the chat. We don't have time for questions today, but I understand you'll be present at the Meet the Speaker session tomorrow, and be happy to answer any questions there.
I'd be absolutely delighted, and please turn up and talk to me. I'll talk to you about anything.
So on behalf of the Society, the conference organisers, and our many attendees around the globe, I'd like to thank you once again for a truly outstanding and exceptionally clear lecture today, and to congratulate you once again on this award. Thank you ever so much.
Thank you so much.